But all right, so what I'm going to talk about is my experiences in engineering and consulting in this area of virus inactivation tools. And this came about. Let me just tell you how this came about. I had been working early last year with a partner of mine on the environmental design of an environmental specs for the MTA, and this is the MTA bus system and the train system. We were looking at we had proposed and we had been looking at building a air particulate sensor, which is interestingly enough another health concern for the city of New York in their bus systems. And they had been moving towards, initially they had been moving away from diesel, and they had gone to natural gas, and they finally had talked about going all electric, and they were in process of moving their fleet to all electric. And Dr. Camille and I proposed to the chairman or the head of the MTA that we actually build a monitoring system that looks at probably the most dangerous of all particulates, which is called PM 2.5, dust particulates. Turns out there's a dust particulate of 2.5 microns or less that's very dangerous, and it's the leading cause of heart disease, believe it or not. It causes inflammations in the lungs and to other organs. So it was a proposal we had been doing, and we had started the prototyping process when all of a sudden COVID-19 hit. And so the director of the MTA says, we've got to change gears and we've got to go to an immediate problem. We've got a virus problem. And at that time, they realized they had a problem. They had zoomed in on the subway system as a vector to spread COVID-19. And so there were couple papers out of MIT that said, hey, your super spreader geographic location is the subway system. So they pulled us into those meetings. So we had a couple consulting meetings with the MTA, and they went off, and our suggestion was on the first slide, Dr. Camille and I put together this system on the bottom, to the bottom left. On this slide here, you can see there's an ultraviolet C-band emitter that we had suggested that we use this on the, at that time it was the bus system and the MTA. And so we had contacted also the governor of New York and said, hey, this system has been proven in hospitals and I say in here to kill viruses, but you can't kill a virus. It's not a living organism. What it is what you do is inactivate the virus. So technically, this is incorrect in that we weren't going to kill it. We were going to inactivate it. And so in doing the research for this, since the mid-20th century, hospitals have used UVC for OR room. So then they want to kill viruses or inactivate viruses and bacterium and fungi and other sorts of parasitic infections. They would leave a 400-watt tube UVC 254 nanometer to sterilize the OR room between surgeries and in the evening and there's no person that can be present for reasons I'll get into here. So we took a standard technology and we said, this is how we can deploy it in the subway system. And they kind of ran with it and contracted with a company. Let's see here. All right, I'll get to that part. So they contracted with a company called PURO and they extended the UV system through PURO to go and have PURO install. And then they contracted with Columbia University, Dr. Brennan, who a person I know, who they paid Columbia University to monitor the progress of the MTA subway system, and the progress meaning the effectiveness and progress in terms of virus inactivation is measured as log reduction. And we'll get into that in a little while. But so right after we made that proposal back in March, we gave a formal proposal. This report from the Department of Homeland Security came out, and this was interesting to me because we have ways of measuring the spectral light output, but we have no way of measuring the ultimate effectiveness of virus inactivation. But who does have Department of Homeland Security has a laboratory called the NBACC. That's a full biocontainment laboratory in the US that they can do some testing. But for some strange reasons, they said, look, we think there's three factors. There's three factors that's causing or modulating the virus load. And basically the three factors are temperature, humidity, sunlight, and they looked at some chemical disinfectant that they in this condition in a biocontainment laboratory. So they used real viruses, which is something I don't have a wet lab, but I do have ways of measuring a various spectrum of UV light, and I have all the spectral equipment and spectrophotometer to actually measure the wavelength and so forth in light and what's ambient. So what they did, is they simulated sunlight and they mixed the right concentration. I say the right, but to simulate sunlight. And there's some issues in doing that. But let me just jump to their takeaway from their testing. And they're assuming you have aerosolization of viruses in air, and that can go into a surface. It can stay in the air, it can go into surface. And then the host is susceptible through the sinus and lungs. And that's how COVID works, by the way. If you look at the design or the way it infects or it dominates cells as it goes into the sinus and attaches itself to tissue, and then it can inject itself into the DNA load into the cell. So what they did is they put together aerosol chamber to control temperature, humidity, and sunlight simultaneously, and then through individual variability, keeping everything else constant. So the bottom line here is in their chemical testing, they started with bleach, hypochlorite solution, and isopropyl alcohol. And then there's the takeaway was, this is limited in effectiveness because, this is manually applied. This isn't sprayed bleach or anything like that. So this is, they're both effective in disinfecting both old, dried, and freshly wet contaminated saliva on a non-porous surface. So that was their chemical conclusion. And it talks about percent. If you look at the matrix on the left, you've got the type of droplet and the chemical, how effective it was measured in percent. So if 70% isopropyl was found to be effective in a wet 99.9, dried was 98.1, and so forth. Diluted bleach was found to be more effective, but that's after a 5-minute contact with diluted bleach. But obviously that's limited. The first approach on the subway system and the entire MTA was to take cleaners with gloves and diluted bleach and go around and actually clean the surface with hand scrubbing the rails and so forth on the subway. But here's an interesting result. So this same Department of Homeland Security biolab, they tested it with sunlight, and they found it to be also very effective with virus suspended in saliva treated with full intensity stimulated sunlight, rapidly killed virus and aerosols. They plotted the half-life of roughly 2 to 3 minutes at room temperature. 90% decrease in virus, and this is talking about the virus load or concentration in 9 to 10 minutes. Without simulated sunlight, they detected no loss. So it showed that sunlight increased the inactivation in aerosols and in surface also. So their takeaway here was outdoor daytime environments are lower risk for transmission, which to the audience listening to me talk through this, this report came out in early 2020. So why are we in a lockdown? Why do people stay indoors when we see an obvious report from the Department of Homeland Security saying it's much more effective being outside? So, and I think you see that health clubs and so forth have moved outside, and you've seen in some areas classes have been moved outside, and TCNJ is proposing more activities under outside. Okay, so ongoing work, they're talking about testing with additional solar and relative humidity levels. Now, these are my notes at the bottom here, is UVB is not as effective as UVC, but it is emitted by natural sunlight. 95% is filtered out by the ozone layer into the atmosphere. So this is the maximum UVB irradiance near the irradiance is a term we use for amplitude and flux amplitude. Near the equator is about 250 microwatt per centimeter squared. So we know from the studies done that UVB is effective only in long periods of time of exposure. And so they're talking about what they actually measured in simulated sunlight with 5% UVB was pointing into the right direction of using ultraviolet light. So that was the area I had focused on especially after reading this study. It said that 5% of UVB made a difference in risk for transmission and protection of staff. And the other conclusion there was increasing air exchange rates indoors may reduce, and since then that's been proven to be effective, increasing air exchange rates indoors. And that leads into one other area I'm going to talk about in a little while, which is the combination of air exchanges. Air exchange is a name for a system that actually is an actual blower system that moves air through indoors. And which includes there's air exchanges that are on subway systems and in homes and in basically air systems. Businesses have air exchange systems. So one of the thoughts I had when I read this was, and again this was early 2020, was to also combine motorized air exchange with UV systems. And that's something I'll come back to in a little bit. All right, so fast forward, the subway system, what they determined to do was to sign a contract with a company that supplies UVA, B, C. So we call this a broad spectrum light emitters. And the New York City subway is actually it's still continuing. The pilot continues from 1 in the morning to just before 4 a.m. when they shut it down. Now, why do they do that in the middle of the night? Well, there's one little problem with ultraviolet C-band, especially. It's very dangerous to human and to any living creature or human, because what it does, and we'll go into how it works in a minute, but it's also a potent generator of cancer carcinogen. Ultraviolet light is used in laboratories to generate cancers. So they use it in hairless mice, for example, as specimens of reproducing cancer. Instead of humans, they obviously use mice and other animals. But anyhow, so it's very effective to introduce cancer. And the same mechanism that changes the DNA, that modifies the DNA, is the same mechanism that causes cancer. So you don't want any living person to be sitting in there when these bulbs go off, basically. So that's done in the middle of the night on the New York City subway system, currently. And I mentioned a Professor Dr. Brenner of Columbia is actually testing the effectiveness. They're in complete lockdown, even though I was a consultant in the initial design of this. They somehow don't want the word how effective or how little effective it is. They don't want to make a press announcement on this yet. So it's still undergoing analysis at Columbia on the results of the test. But I can tell you, it definitely is pretty effective according to my testing. And I'll get into that in a minute here. So the system they used is manufactured by a company called PURO, Violet Defense. And what it does, it's a pulse xenon tube source. Now, what that produces is radiation from 190 nanometers to just over 400 into the visible light spectrum. So it's a pulse that actually generates visible light all the way through a short UVC spectrum. And this is PURO is the manufacturer. Violet Defense is the patent holder, which is the research that I was able to get from looking into this. And also we through company I'm consulting with right now, we have a contract to distribute this system right now. And okay, so I've been doing research for a company called Swemco in New Jersey, and this is what we've been working on. Violet Defense has patented this pulse xenon technology because it comes with the special lens that, basically UVC is not passed by glass. Glass will block UVC, so you have to have special plastic form lenses that work at a higher temperature and so forth. That's included in their patent, is the lens system. They call it Violet Defense Surface and Air Germ Elimination, or SAGE UV products. And like I say, it basically generates UVC-B and A and violet blue light. What I'm going to do is, let me jump forward. I think, let me just talk a little bit about, all right, let me just talk about effectiveness versus wavelength. So this is, if you look at the x-axis, it's 200 nanometers to 300 nanometers. You could see where it basically scrambles DNA and RNA. That's the dotted graph, most effectively at close to 260. And if you look at it, 253 is what they've been using for 40 years in the pulsed tube systems. And in this xenon gas systems, they have a broad spectrum, but you could see the effectiveness is in the 253 nanometer area, where you have the peak in the dotted black line, and that's for DNA and RNA. And So here's how it works. Let me just dimertization. All right. So here's what happens if you have the virus. The virus is just a wrapper that has RNA or DNA around it, and that's inside of it, and then you have a wrapper around it, and you expose it to UV radiation, and you have what's called the thymine dimer gets created. So if you look at the DNA structure on the right, you have what happens is it's fused together. Basically, your genetic code is screwed up. So you cannot reproduce. This doesn't reproduce well once the dimer has linked the thymine together. All right. So you see the bond is broken and it's fused, so it's damaged. So the UVC photons cause the critical damage to the genomic system. Nucleic acid and microorganism proteins. So this is the sequence that consists of the nucleic acid that's out of order. If it's out of order, it cannot build a structured protein, which is the product of any DNA and RNA. So it prevents them from replicating and surviving where the adenine-thymine bond is collapsed. It's missing, and then it bonds directly between the two adenines. Now, this is what's the damage that it does. And let's see where I get, let me go back up here. And so this is the lamp. So this is the actual light or lamp system that's used to produce this inactivation. They have the low pressure UV, that's low pressure gas UV lamp, medium pressure, and then they have the far UVC lamp of 222 nanometer. It's called far UV because 222 is much shorter, much higher frequency, shorter wavelength than a typical wavelength we use. And come back to that. 270 nanometer is a convenient spectral from the new ultraviolet LEDs that are being prototyped and out right now. So if you look at the bottom, this is what's caused by various lamps. You have the various spectral peaks. And the far UV was pioneered also by Columbia. It came up with data saying that 222 nanometer actually protects the human cell. It doesn't cause the same problems, mutations you have in humans that you have with the longer wavelength. So that's one area of research right now that Columbia's been doing for the last 5 years, but they're testing, they're still testing. They didn't get FDA approval, so right now it's not a commercial product, but they're saying that, the ultimate, what you're really looking for is a solution that can be used with people present, like people sitting on the subway cars, and then actually have the optical radiation being constantly, and we're going to get into that idea too. So let me come back here. All right, so back to the PURO system. So this is this box here on the left, and there's two of these light sources per subway car. Each car has 2 PURO light sources, and you can see when there is a visible spectrum. So when they fire once, typical default programming of the electronics. Now, since the electronics need to check to make sure there's no people that walked in, there's actual sensors that determine if there's a person or an animal present, and it shuts down the unit immediately. Like, for example, if the doors accidentally, or they're locked, but if they're open and somebody walks in, it automatically shuts off, immediately shuts down the light sources. And this is to protect the humans walking in. Okay, any questions so far? And it's a lot of material here, but it's an interesting pilot. There's quite a bit of research that went into this. And so looking at the spectrum, so I actually have these PURO systems and I've deployed them in some areas. So the spectrum analyzer that I use for that takes these kind of images. So it shows you that this is after a certain, after 190 nanometers, this is its broad spectrum. So you have all this energy that's being generated by the Xenon, pulse Xenon technology. And then you have the visible spectrum that's also being generated. So if you want to see something really eerie, you should see some churches in the middle of the night when this thing is in operation. It looks like there's actual lightning going on in the church. So the PURO is the broad spectrum Xenon pulse, and the 254 nanometer, which is the sweet spot for inactivating viruses, requires about 2.6 millijoule of energy, which is 2.6 milliwatt for 1 second, because energy is time, of course. So you get 2.6 milliwatts for 1 second, or 87 microwatts for 5 minutes to eradicate 100%, or is it 98% of a MRSA population, which is bacterial. One of the testing, like I say, is difficult. There's only one type of virus that you can actually test and be safe handling and dealing with, and that's called a phage. So phage is a virus that you can treat like a virus, except it doesn't attack people. It doesn't. A host is not a person. A host is another bacterium. So a host is a bacterium and not a human or an animal. Okay. So this is the sweet spot, 254, but you're generating all this other radiation that I have their white paper. They go into actual testing of the different levels of energy. Now, where my testing stops is where I have the spectrum equipment to measure the output. So if I put this outside, for example, on a bright sunny day, I can see a nice spectrum of UVB, but it's severely attenuated. You know, it's 5% of what the sun's producing. That's mixed into sunlight and then gets attenuated by ozone. All right. By the way, ozone is another powerful tool for the elimination of or the inactivation of virus. So that's another system, except it is also very dangerous. And people that are asthmatic, it can kill you basically on high dosages of ozone. So ozone doesn't come up a lot in the research. And you see quite a bit of research in food and in water too, because you have the same problems of viruses and bacterium and fungi in water. So you really want to clean that out, and then use the same kind of ultraviolet system for water treatment. Any questions so far? Okay, so that's the PURO system. We covered the original. The system they use in an OR room is not a Xenon system, but it's pulsed 400-watt gas tubes that are optimized for 254 nanometer. Get this in the screen here. So what we're looking at here is an example of how if you have a point source, a point emitter, And I got to shrink my screen here for a second. Okay, so S is your light source, and R is representing your measured points. All right. So this just illustrates it's a square law. So you have to compensate for your distance. It drops off dramatically as you go in a distance from the point source. So a couple ways of solving this problem is one way is to have multiple emitters. In our case, we're using multiple light sources, multiple pulse xenon sources to produce less of a point source and more of a plane source. And the other solution is we get closer. I mean, if you can change the distance from your seats on the subway or the rail on the subway, it's much more effective. So this is one area where robotics plays a big role, because the idea is we have a patent with Swemco. I have a patent that actually changes. It's a linear robot that actually takes the emitter and takes it right up against the rails and the seats of a subway car. So that's in right now. That's been filed, but let's see what happens. All right. Let me see if I can. The screen is expanding beyond the text, not showing up the text of the thing. Let's see here. So control. I have a wildcat here. Let's participate. Oh, all right, kitty. Beat it. Get a little obnoxious. All right, so backing up a bit, we're looking at virus inactivation, and what are the size of the particles that we're talking about in terms of droplets. So it's basically covers a wide area. The virus itself is about 3 microns, and it's so it's very small, and it's aerosol. So when you have contaminated air and surfaces, this is what you have to address. And let's see here. So depending upon whether it's on the surface or it's in the air, some of the newer techniques, let me lead into this here. Let's see. All right, so let me talk about some solutions that are brand new that just literally came out on the market within 30 days. So General Electric, division of GE, my alum, is working on a very advanced system that's the size of a carbon, it's actually a little bit smaller than a carbon monoxide detector. So what they said is, what really is, can we quantify the risk to a human? And the answer is yes, IEC 62471 spec, which is labeled the photobiological safety of lamps and lamp systems. So you basically have classification of 3 hazards due to ultraviolet light. Actinic, which is skin eye exposure to UV, can lead to eutherma or sunburn, which leads to cataracts. And the near UV, which is directly induces cataracts into the eye. The first one is a thermal effect, the actinic UV hazard. The near UV hazard is the cataract concern, and the blue light exposure, it results in photoretinitis on the retina. Now I'm not an optometrist, but anyhow, so that they're the 3 risks that they've quantified. So GE Current, a division of GE, said, well, why don't we take the studies? Now, there's a whole series of studies that's been done for the last 10 years, mostly in the area of tuberculosis, which is to put the ultraviolet system they call upper air ultraviolet system. So you mount it at a considerable distance and you control the level. And you keep the level low enough, 1 second here. Okay, so the idea is to keep the level low enough so you end up, okay, so you end up basically not having the, 1 second here. Okay, where was I here? Okay, so these three risks are quantified in IEC 62471 in the spec. So GE said, let's just go underneath this level of energy, and where there's not going to be an actinic or near UV or blue light hazard, let's keep the energy down to where it's safe, where it's within the spec, IEC 62471. So they have a small device, it's LED based, that's generating the same, near the same frequency, that's the magical frequency of 254 nanometers. And in some cases, it goes into the UVA. So GE has both the 254 system and the UVA 365 nanometer LED system. And again, it's out on pilot. I don't know if it's in production yet. It just went into pilot mode. So the 254 emitter, LED emitter, they know what that will do, and they know the radiation. So they could source the level into tables of log reduction, so they can tell whether that's effective or not. And 254, as I said, we have over 40 years, almost 50 years of support on that. It's not visible, but it also kills bacteria, mold, fungi, inactivates viruses. And then UVA takes a longer period of time to inactivate. It's minimally effectiveness on virus. Okay, so then because this is a dosage system, they said, okay, well, if it's safe now to humans, we want to leave it on all day on a light dark cycle, which continuous means 8 hours on, and not have any switch off if there's a person underneath, because it's fine. You know, you're within the killing a virus and not inducing cancer in humans. Anyhow, I have these units for test. Anybody wants to volunteer, which I would have to set up an IRB. But right now, what I'm doing is I'm taking this particular light and I'm looking at the intensity. It's designed to be installed at 11 feet. 11-foot ceilings. They're calibrated depending on your ceiling and how you're installing it. So again, at the correct intensity, both UVC and UVA can meet the IEC limits for human exposure. So this is all really interesting stuff. Now, I've talked to the engineers at Motorola who designed this system, and they're saying, you know, it's perfectly safe. But now they have a marketing problem, which is convincing people these are safe, and start installing it throughout the school systems and all over the world. So that's where we are with this. Let's see here. So where they're getting a 2 to 4 log kill is over 24 hours for viruses. So again, they're saying this is going to be upper air. Somebody coughs, it's going to take a while to completely inactivate the viruses because it's lower power, longer exposure time. And this is what the units look like. The one on the right is the one I've been testing. That's the UVC unit. Very, very tiny, very nice. And I'm using that with my spectrum analyzer, my optical spectrum analyzer. And right now, there it is again. So here's again, we're back to the time. Aerosol viruses take roughly 3 to 60 joule per meter square to inactivate 90% pathogens. Viruses on surfaces or in suspensions require a much higher dose to inactivate in the word. I can't read it very quickly. So if you want to do this very quickly, you need to go with the higher levels of UVC. So this is the most important thing for me, which is what I'm designing for, is an IEC 62471 compliant dose of 254 is 60 joule per square meter per day. And again, a day is defined as 8 hours. For a virus with D90 of 3 joule per meter squared, 99% reduction happens every 2.5 hours. And that also complies with the IEC spec. Okay, some word about, there's a lot of claims in this field that's not backed up by any testing. This particular robotic delivery system is not viable because if you do the UVC output and exposure time required, you need very large batteries. And I could share a system that's a robotic system, and they're not small, they're not petite, and so basically these don't work the way they're designed right this minute. So if you see these for sale at $19.95, don't buy it. Okay, let's see how much time I got. Any other questions? Any questions at all? Okay, this is, like I say, so this points to success on the using the GE system, which is reduced power in the ceiling, coming from the ceiling, and basically leaving it on 24/7 or 8 hours minimum per cycle, 8-hour cycle. And so this is something, like I said, I'm testing. Right now it's in a warehouse environment with 11-foot ceilings, and I'm measuring the energy. Then what I do is I'll take the energy readings at 254. Now this is pure 254. The PURO lights is broadband, but this just has a nice signal that comes out of the noise. So right now I've been playing around with integration to bring it out of the noise. It's pretty much, it's very low level, and that's what it's designed to be. And so then I correlate the level with the power output with the tables of log reduction on COVID-19 viruses. The other option would be to contract with a wet lab and do some phage testing also with this. I have no way of accessing Department of Homeland Security's laboratories, but it would be interesting if I could get their access to their lab. Okay, okay, Shane, that's pretty much what I got. If any questions, you can.
There's one in the chat. I can read that for you. It asks about like the use of UV phone sanitizers. So it's just a little like pod that you stick your phone in and it blasts it with UV light for a little bit.
Yeah, I haven't tested those, but they're low level, and so they're safe. You have to close the case when you put your phone in it. So I haven't tested those. I understand there are some tests out there. I did read some tests where some work and some didn't. So some were not even UVC. Some were just a light source. They figure nobody's going to know the difference. But whatever you get from at cheap prices, I think they're down to $19 now, the ones that sanitize your phone. But we're looking at industrial strength. The PURO lights themselves, and we have customers for them, they're $6,000 for a dual-headed tripod system. And we see a lot of interest in churches and in synagogues and in funeral homes. And I don't know what the school system's pretty much, I don't know. It's hard to get to who's in charge, who's buying anything in a public school system anymore. I don't think there's a budget to buy anything, but it certainly points to you know, sunlight also being useful, being outside in the sunlight and exercising, and there might be a cofactor with sunlight, because sunlight and UVB is how your body generates, manufacture the hormone D3, vitamin D3. It's not a vitamin, it's a hormone, but D3 is what's being produced by the little bit of UVB that you get in sunlight, the 5% that gets passed through. So there is this correlation, maybe, I don't know. The data is being worked on right now. There's another concern too with the transmission of viruses through other means, like through dust particles. When I started the research with the MTA working on dust particles, and lo and behold, I found some research medical papers out of Taiwan that's saying these can be transported on 2.5 micron dust particles. So a conspiracy-minded person I am, so I'm thinking, that's not a good idea. I've never heard about that. But if you wanted to transmit these viruses across continents, you can do that on very tiny, non-visible dust particles, which if you're in the bioweapons business, it might be interesting, or for protection. So on the other hand, this ultraviolet light works on all viruses and bacterium, and anthrax too. Also works on anthrax. Usually for anthrax, though, they use a higher energy source than UV. They use deadly gases to eliminate anthrax and seal up the post office and wherever they think there's, you know, whether they have a contaminated area, they use a gas to contaminate it. All right, any other questions here? I think this is some good material for grant money too. I think we're going to try to get some grant money. Hopefully at TCNJ, it sounds like it should go in the BME department. I've talked to Chris, the chair of the department, and I know he's interested in this work. All right, that's about all I got.